starting my fourth year at NYU, PhD in computer science. I'm working with Andrew Gordon Wilson. Sam actually had the same advisor as me, but graduated, like a year or two ago, and is now at Prescient Design. So yeah, let's get started talking about the paper. So even though we kind of develop a pretty general methodology in this paper, we focus very specifically on antibody engineering. And just to provide a brief, introduction to antibody engineering, antibodies are these complexes of proteins, and they have a constant region and a variable region. And these variable regions contain these smaller regions called CDRs that define a binding interface with an antigen. So the antibodies bind onto these antigens as a way of signaling to the immune system. And the antigen tends to have a site called the epitope that the antibody binds with a paratope. And typically in antibody engineering, kind of traditional antibody engineering, a human will just tweak these CDR regions to try to improve binding or change other properties of the protein. So we basically want to develop a more general ML methodology to do this. And we kind of frame our particular approach within the framework of adaptive generative design, where we start with a pool of proteins. Each of these proteins has a set of labels, multiple objectives, and over a series of rounds, we're going to try to optimize these different objectives while trying to be very efficient about the amount of evaluations we need to do, and through generating mutations from generative models and ranking things with discriminative models. Okay, so in this kind of framework, which we're participating in, there are a couple different pieces. One, we're going to maybe come up with discriminative models that are going to predict the different properties that we care about. And we have a proposer model, which is like a generative model that's going to propose mutations. And we have external ranking models that are going to pick the mutations that matter. And in particular, this way of picking things could actually be based on uncertainty, for example, so that we explore the space broadly. We don't just exploit the things that we already know are good. Or it could be based on combining multiple objectives so that we make sure that we get proteins that do all the things we care about. So the most naive way that we might conceive of doing this kind of adaptive generative design is basically just taking a huge library of proteins or mutations and training a surrogate model to predict the thing we care about, and then just ranking the mutations or the library based on this surrogate model. If we want to be a little bit more complex, we can have this kind of closed loop where we're constantly proposing new things, expanding that library with a generative model, and then labeling things with a surrogate model and retraining the generative model, often doing actual experiments in the lab by labeling things in the lab and using that to retrain the generative model. And the piece of this that also is important here, because our laboratory experiments are quite expensive, is making sure that we are prioritizing points that are going to give us a lot of information. And that means that our acquisition function here, the thing that helps us choose the next points, is going to take into account not just what a good point is, but also the uncertainty around that point. So our contributions in particular focus on this model-based optimization framework. We basically develop a methodology for proposing points that are going to simultaneously maintain the plausibility of a candidate while optimizing its fitness. So most naively, you could imagine a framework in which you have a starting set of seed sequences, and you train a generative model to sample within the neighborhood of those seed sequences. You can imagine kind of an unguided generative model. And once you sample a lot around the neighborhood of those seed sequences, you can then just take the best points and return those. And what our method is going to do instead is directly generate things that are both good in the neighborhood of what we started with, but also improved in their fitness values. And this is going to be our method called NOS for generating guided sampling. So we have two core contributions. First, this gradient guidance method for sequence diffusion models called NOS. And then second, we introduce this potential that we're going to use for our guidance, which allows us to do multi-objective and uncertainty-aware optimization. So I'm going to talk mostly about the first part, and then Sam is going to talk mostly about the second part and how we leverage that to do actual antibody lead optimization. So yes, let's start with NOS, the guided discrete diffusion part. So as I mentioned before, the most basic approach maybe to this adaptive generative design is to train an unconditional generative model, use this to sample within the neighborhood of some starting seed sequences, and then just rank those with an external discriminative model and pick those as the points to use in our next wet lab experimental trial. And there are a couple approaches to generative modeling in this way. Broadly, there are two. One is structure-based and one is sequence-based. So in structure-based methods, the stochastic sampling happens in structure space, so in 3D coordinates. And in sequence-based methods, the sampling happens directly over amino acids and the discrete identities of those amino acids. So to apply structure-based methods, generally we have to go from sequences to structures, do the sampling in structure space, and then get back to sequences, because ultimately we need the sequences in order to make the proteins. Okay, and that's opposed to sequence-based methods where we basically are staying within sequence space the whole time, because we never needed to apply the sampling procedures in 3D coordinates. So often people favor structure-based sampling approaches because structures have very strong physical priors over possible configurations. So for example, there are like certain physical properties that cannot be, for certain configurations that are just simply not possible, and that allows us to enforce very strong priors over the possible samples. Structures are also quite desirable because in a sense they're more uniquely determined than sequences. Multiple sequences can correspond to, multiple sequences can correspond to one structure. And of course, structures are in a sense the determinants of function. So of course, structure is a kind of more intuitive way to think about the function of a protein. Also, from an optimization perspective, structures are convenient because they are continuous objects and thus have continuous optimization surfaces so that we can use gradient-based methods. So because we're talking about generative models, the continuous nature of structures is also quite convenient because it maps more directly onto methods used for images, which are also often treated purely as continuous objects. So for example, the kinds of diffusions that were developed for images are often mapped fairly directly onto protein structures in a couple papers that are very prominent in the community now. So why might we not want to use structures when we're doing this generative modeling task? Well, structures can be quite challenging for a couple reasons. One is that finding structure data in the wild is often quite challenging. Finding a co-crystal structure for a given antibody-antigen complex is quite challenging often. And using folding models to approximate this is often quite limiting as well, because they're not always sensitive to individual point mutations. And particularly loop regions tend to be quite floppy and hard to predict with a good folding model too. And the loop regions, as I described earlier, are what's often the most important for things like antibodies. On top of all this, we need to get back to sequences eventually. So if we're going to use a structure-based method to generate new sequences, we're going to have to use inverse folding models to get back to sequences. And inverse folding can often be quite challenging or generate sequences that are not very plausible. So in this paper, we're going to advocate primarily for using sequence-only methods. for a couple reasons. One of those is that we can observe that because sequence informs structures and structures inform function, much of the information about function is actually already contained within a sequence. So therefore, we can use sequence to function predictors to perform a lot of the tasks that we might care about. And ultimately, sequences are what we want to synthesize, and therefore we should care the most about generating plausible sequences that are likely to express and likely to be easy to make. So within the space of sequence models, often called language models too, there are broadly two families, and I'm just going over this to motivate why we chose diffusion models of the two families. So there are autoregressive models and non-autoregressive models. Autoregressive models you're all quite familiar with probably because of ChatGPT. All GPT variants are autoregressive models, but there are also non-autoregressive models, and non-autoregressive models are often used either for representation learning or for generation. In representation learning frameworks, they're typically used with a single corruption rate. So a sequence is corrupted, and then it's tried to, the model reconstructs this from the corrupted version. And in a diffusion setting, there are often multiple levels of corruption, and the model learns to reconstruct the object at every level in order to sample a new object. So this is just one example of an infilling type task in a non-autoregressive framework. This is particularly convenient for things like protein design because essentially infilling tasks like those introduced in non-autoregressive language models are directly analogous to protein design tasks, because protein design often involves essentially proposing new tokens to replace existing ones at non-contiguous regions that are often quite small. So diffusion models for sequences often involve essentially having a schedule of corruption rates from 0 to 1. One simple example is a mask-based diffusion. We can imagine this sample string here. We're going to have the model reconstruct this after it's been noised with mask tokens at every possible fraction of masking tokens between 0 and 1. And during the training process, we're going to provide it with the mask tokens and have it reconstruct the unnoised version. And then during the sampling process, we're going to start with a fully masked string, and we're going to slowly turn this into an unmasked string, which will ultimately be a sample from the distribution that we care about. And in order to both generate sequences that are plausible and sequences that optimize objectives that we care about, we're going to have two pieces. We're going to have a generative head, which is going to take the masked sequence and generate the unnoised string. And we're going to have a discriminative head, which is going to take the masked sequence and generate the labels that we care about. So in diffusion land, there are also diffusions that operate on embedding space directly. So instead of masking the tokens, we can also apply something like Gaussian noise directly to the embeddings that correspond to those tokens. So there are a couple models that were introduced in this form about a year ago, and we really just want a procedure that works for any kind of diffusion that you would possibly want to use. In the case of noisy embeddings, it looks very similar to the other case. Instead of basically taking in this masked input, we just have corrupted embeddings of some form. And in the generative head, we're going to predict the denoised sequence from the corrupted embeddings. And in the discriminative head, we're going to predict the labels from the corrupted embeddings. And when we use this model to perform infilling, when we go and we train some discrete and continuous diffusions, here we're going to call the discrete diffusions NOS-D for our method, and the continuous diffusions where we apply Gaussian corruptions on top of the embeddings, NOS-C. When we go to actually perform infilling with these models, we see that they tend to be quite a bit better than competing approaches to sequence modeling and also structure-based methods. So we see here RF diffusion and diffab are models that use structure information and are therefore often trained on different or more limited data than our models. and IGLM is a autoregressive model. And we see that the infilling, as judged by sequence recovery, is often better for our models when we apply it to the CDR regions. So, but what we really want is not just, this vanilla infilling. We want to guide this under our objectives. And it's naively actually a bit challenging to envision how we would do this directly over token space, because tokens are these discrete objects. So we can't directly apply gradient signals from our discriminative head, but we can use an insight from the general NLP literature, a prior work called plug and play language model. Basically, the observation here is that we need a continuous latent space, like with structure models, to apply the gradient in. And conveniently, we can see that the activations of the network itself, which are shared here, we can see between the generative and discriminative head, can serve as this continuous latent space. So basically, when we are sampling reconstructions with our diffusion model, we can bias the sampling of those tokens with our external attribute model. So here, our attribute model is going to predict the kinds of protein quantities we care about. But in this case, we can imagine, writing a restaurant review. Our attribute model is positive sentiment, and this is going to bias us towards saying things about the dishes that are positive. So when we combine plug and play language model style sampling with diffusion models, it looks a bit like this. We have a generative model head, as I described before, that reconstructs. We have a discriminative model head that's going to predict the things we care about. And now to sample from the model, we take steps in the activations of the neural network corresponding to three things simultaneously. One, we're going to have a KL term that makes sure that our reconstructions under the guided sampling stay close to the reconstructions under the vanilla sampling. That's this first term here. And then we're going to have a second term that makes sure that the tokens that we sample correspond to maximizing the objective. So we're going to take gradient steps under the potential that we're trying to maximize. And then lastly, we're just going to do some stochastic walking around this latent space. So why use the KL that we chose here, particularly the KL between the guided head and the vanilla head? Well, we can view this as maximizing the LaMBO and latching onto the mode that's going to maximize the objective. So instead of basically vacillating between two modes that might correspond to likely sequences in order to optimize this objective, we want to just collapse onto one of them, the one that corresponds mostly to maximizing the objective that we care about. Okay, so what happens when we go to actually apply this technique? Well, in most of the results in this paper, we're going to present Pareto frontiers, either because we care about multiple objectives or because we both care about optimizing the objective and maintaining likelihood, because of course, in the end, we want to make sequences that will actually express. And here we present the Pareto frontiers between two toy objectives and likelihood on the x-axis. So as our baselines here, we have a structure-based method, RF diffusion. Basically, we're sampling new possible structures in the neighborhood of our starting structure, which we obtained with a folding model. Diffab is a joint sequence structure diffusion model trained on antibodies. And PPLM is using PPLM, this vanilla autoregressive formulation of gradient guidance on top of an existing autoregressive protein antibody model. So as we can see, our two diffusion models tend to push this Pareto boundary outwards, which shows that basically we're good at both optimizing the objective and maintaining high likelihood at the same time. So that's kind of exactly what we designed the model to do. And it's good to see that with in silico objectives, that's exactly what we get out. So takeaways from this section, we're going to move into Sam's section next, are that diffusion models are great for infilling. They generate high likelihood sequences with good sequence recovery. And also we can use NOS, this gradient-based method, to generate sequences that both optimize the objective that we care about while still having high likelihood. So in the next section, we're going to show how this method can be used in tandem with a more comprehensive potential to do the kinds of things that we care about in protein design, which is multi-objective optimization, uncertainty-aware optimization, and we're going to show how we can use this to design actual antibodies. All right.
Cool. Thanks, Nate. You want to answer the question in the chat while I get my slide sharing set up?
Oh, sure. Yeah. I don't think there's a question in the chat.
Yeah, there's basically the question was,
oh, I see it.
Was the baseline RF diffusion plus protein MPNN, which I believe it was.
Yep.
Another part of the question was, did you allow sequence lengths to change? And then the other question was, did you try essentially just diversification with protein MPNN in a fixed structure?
Yeah, so the answer is yes, this was RF diffusion plus protein MPNN. And the answer is I tried both variable length and fixed length, both formulations. And because basically both are possible within our framework as well by using alignment tokens. And the last question was, did we try MPNN on its own with a fixed structure? So basically the way that I set it up was that we sampled a bunch of variants of the structure. For each of those variants, I sampled variants of the sequence with protein MPNN. So there's variants coming from both modules. I didn't kind of like disentangle those sources of stochasticity, but yeah, if that answered the question.
Cool. Thanks, Nate. Right. So Nate covered a lot of the, I guess, general details of our method that are really not that specific to antibody design or even protein design. Like if you were going to try to design some other discrete sequence, essentially all the things that he just mentioned would apply. For example, if you wanted to do like gene regulatory element design, it's essentially just a different vocabulary. And so what I'm going to focus on in this section is some of the kind of more specific challenges that we encountered applying NOS to antibody design and what we did about them. Right. So one of the central concepts that we're going to spend a lot of time on is what our acquisition function was. And for those of you who are familiar with Bayesian optimization, essentially an acquisition function is derived by defining a utility function that measures, essentially how much you value one sequence over another. And there's a couple considerations that we need to have. The first is that antibody design and drug discovery in general is very multi-objective. There's multiple properties that you care about. Certainly one is expression level because that determines how much you can make. Another is binding affinity. If you don't bind to your target antigen, that molecule is kind of dead on arrival. And then there are various other properties like aggregation, oxidation potential, and other things that affect things like shelf life. And one of the characteristics of this particular problem is that the assays that we use to measure these properties are sequential. So if a protein doesn't express, then you won't get to run anything else because you don't have any of it. And if it doesn't express and bind, then you won't run any of the downstream experiments because it's just not worth the time. And so essentially that means that you have this hierarchical structure and a lot of missing labels. There's also multiple rounds of design and observation. And so you need to account not just for like how good you think this molecule is right now, but also account for the fact that there's some value of information if in observing molecules that you're uncertain about. And then finally, the assays that we use to measure these properties are extremely noisy. And so if you're using like improvement-based notions of utility, then you need to account for measurement noise when you're estimating the improvement of some hypothetical design over what you've seen in the past. And yeah, so just as kind of a brief overview for any of you who are not familiar with multi-objective design, because this is going to be one of the key parts of our utility function. So in the case of single objective optimization, all optimal or non-dominated designs are essentially equally good. What it means is like you're not going to differentiate between these optima without some additional information. That's essentially what it means for a problem to be single objective. In the multi-objective case, now non-dominated designs can be differentiated, and they're differentiated along this Pareto frontier where they represent different trade-offs. And so the question is, how do you measure the value of new designs when you have more than one objective? Because you can't just rank them. And so what you're going to do is you're going to start with some baseline solutions. These might be your molecule leads, for example. And they're going to have some functional properties. And you'll estimate them here. In this case, perhaps maybe you use the labels from a former experiment. And essentially what you're going to do is you're going to choose a reference point, and the volume enclosed by your designs in this reference point defines something that we call a polytope, and this polytope has a hypervolume. In this case, in 2 dimensions, the hypervolume is just this area that's shown in the shaded region. And so now the question is, okay, how do we use this to get a notion of a best candidate when we have multiple objectives? And so what you're going to do is you're going to use a model to predict the properties of these blue candidate designs. And then you're going to ask yourself, what would the volume of this polytope be if I added each particular blue point? And that'll allow you to essentially choose the point that maximally increases your hypervolume, which turns out to be equivalent to essentially pushing out your Pareto frontier as much as possible. And so hypervolume improvement is just the difference of your hypervolume after adding this new point minus what you had before. And to address uncertainty and the exploitation trade-off, which I alluded to earlier, essentially we're going to make use of Bayesian probabilistic predictions that encode some notion of epistemic uncertainty due to lack of data. And so what that ultimately will reduce down to is you'll draw samples from a posterior. Each sample will have an associated hypervolume improvement, and then you just average across the samples to get what's called the expected hypervolume improvement. And there's one additional adjustment that you make for the noise in the observations that I'm not going to cover in detail here, but essentially what it boils down to is you have to use your posterior to estimate both the location of the baseline points and the design points. To address the hierarchies, we're going to make use of what I would say is a trick you may have seen before in when modeling biological data, is this notion of zero inflated modeling, where essentially you kind of break these properties out into a binary constraint, like did the assay succeed or not, and then a continuous value that is the measurement of that assay conditioned on the event that the assay succeeded. And then you'll have this directed acyclic graph that essentially encodes the notion that some assays can only succeed if other assays succeeded before them. And then what you'll do is this will induce some joint distribution over these y's, and these y's will be what is ultimately fed into the multi-objective acquisition function. So this is just as a bit more of a concrete example. So as Nate mentioned earlier, our model has two high-level components, a generative component and a discriminative component. The discriminative component can be further subdivided into different objectives, and then within those objectives, classifier heads and regressor heads. And so first, your classifier will make this prediction of whether it thinks the sequence binds or not, and then conditioned on the event that the sequence binds, then we'll essentially try to do the ranking based on the continuous KD of these antibodies. Okay, so again, as Nate described earlier, to do the kind of guidance that we proposed, we need the discriminator and the generative model to share a latent space so that we can compute gradients with respect to our acquisition value, which is a function of the discriminative outputs, and then use those gradients to modify the output of the generative head. And so in this case, this is essentially what we do. So we have a single network that takes as input the variable regions of the antibody and the antigen sequence concatenated together, along with some utility tokens. And then there's a shared encoder that's, in this case, just 1D convolution residual blocks that then splits out into various branches. And so one branch is learns some task-specific features to predict expression. One branch learns task-specific features to predict binding. And then we have just a linear head immediately on top of the shared representations that predicts the tokens. And then the predictions from expression and binding are fed into the NEHBI acquisition function, as I just described in the past few slides. So to address the fact, I mentioned this at the beginning, but because of the fact that, for example, antibodies that don't express won't bind, we have to deal with the fact that we have essentially a lot of partial observations. We have sequences that maybe have no yield and no binding KD values because they weren't measured, or they only have one, or they have both. And so in order to train a single model on these partially just disjoint data sets, we're going to use multitask learning. And we're going to use a very simple variant of it, where essentially for every gradient update of the model, you randomly sample a task and then randomly draw a mini batch from the set of points for which you have the appropriate labels for that task. And in the case of the generative head, it'll just be the set of all sequences that you want to emulate the distribution of. I think something that maybe is Nate didn't mention explicitly, but at least in this part of the talk, the models are all jointly trained. So it's joint generative and discriminative training, although you can also use it on frozen generative representations as well. So as we discussed before, the acquisition function is this expectation with respect to a Bayesian posterior distribution. And our approach to estimating sampling from this Bayes posterior is going to be inspired by deep ensembles, which I'm sure almost everyone is familiar with. Deep ensembles have a great track record, particularly in industry, as being quite robust with respect to both distribution shift and just kind of general performance and accuracy. However, deep ensembles, the big drawback is that they do not share latent space. Traditionally, deep ensembles are completely independent models, which kind of defeats the purpose of what we've been trying to do with this architecture. And so what we're going to do is something called a partial deep ensemble, where essentially, the first, let's say, k layers in the network are shared. And then when you reach the branching step, you essentially copy those branches. And so you have, in the end, a network where the shared components are not ensembled, but then the branching parts are ensembled. And so that's what we call a partial deep ensemble. We primarily, for this section, we primarily worked with encoder only architectures. And so what that, and the reason to do that is it essentially gives us very fine-grained control over the number of edits that we make and where those edits are going. So if you know, I get told, don't make any designs with more than 8 edits, that's easy for me to do because I can just put a maximum of 8 masking tokens in the sequence. That's all very well and good, but it puts more burden on me to choose where the masking tokens are going to go. And so one of the methodological developments that we made in this paper was to start relying on methods from model interpretability, specifically saliency maps, to essentially automatically select edit positions that were going to maximally improve the model's acquisition value, or the sequence acquisition value, rather. And one of the things that really surprised us was that for smaller edit budgets, which are very common in antibody lead optimization, the effect size of edit position selection is actually much larger than the effect size of like gradient guidance of these token distributions conditional on a specific choice of edits. And so one of the takeaways from this talk that I hope you'll remember is that, you know, if you're trying to do antibody design and you're trying to like you have a molecule that you like and you're trying to like make sure that you're still hitting the same epitope, a lot of your energy should be going into where are you making changes as opposed to like, you know, kind of tweaking token distributions. Because at the end of the day, it's like by a simple combinatorics argument, in it, you know, the variable region of an antibody, there's 300 positions, and you have to choose, let's say, 10 of them. And in the case of amino acids, well, there's only 20. And so it's, I would say, a fairly simple combinatorics argument to kind of see which one is the bigger space that you should pay attention to reducing. So the data sets that we used to train this model, there were essentially 3 types of assay that was done. So one was NGS sequencing, specifically with a vendor called 10x, and that produced what's called paired sorted repertoire sequences. So these are antibodies that were from the immune system of an animal that was immunized with the antigen of interest. And so this is a nice thing to train the generative head on because it's in a sense a very targeted library already of the sort of thing that we want to generate. And then for expression, we had circular and linear transfection data. We used both circular and linear to train the classifier, and then only linear to train the regressor because the linear was the expression system that we were using currently during our experiment. And then finally, to measure binding affinity, we used an assay called surface plasmon resonance kinetics. And we had about 10,000 measurements in total across all lead molecules and targets. So I just wanted to share kind of some results of how our discriminative heads did before we went to, so what this showed is if you fix a certain point of time, train a model and then generate some sequences like with NOS, for example, and then observe those in the lab, how well are you doing at predicting the outcome of that next experiment? So I want to be really clear that this is a very specific notion of generalization. And what you might be surprised to know is that at least in this kind of limited regime where the edit distance is not that large, sequence models do quite well. You know, we were able to get, in the case of binding PKD Spearman rho over 0.6 on a fairly, you know, small data set. So, you know, a lot of times that you will hear people say, oh, well, you know, binding affinity is a really hard problem. And it is a very complex phenomenon. However, when you are, you know, let's say only interested in modeling a certain neighborhood around a lead molecule, you fairly simple sequence models can actually take you quite far. In addition to this KL penalty in the design term that's kind of preventing us from moving too far out of distribution, we also had another kind of safety rail that essentially was a distributional hypothesis test that used engineered features. So this was not deep learning. This was essentially some engineered features plus like a kernel density estimate and a likelihood test. And this was intended to kind of further guarantee that we are in the space where our models utility estimates are meaningful and not just, you know, the being exploited by the optimization algorithm. And so to get to results, just to give people some context, one of the primary prior results that's been published up to this point on sort of ML-assisted antibody design on similar targets was this paper from Absai, Shanasazadeh et al. And what they did was they took a very well-known antibody called trastuzumab, and they did two variants, one where they infilled only CDRH3 and another one where they infilled more, I think it was CDRH1, 2, and 3. And they just used an unguided protein language model to do the infilling. This generated a large library of about 1 million sequences that they screened down to about 400 with a high throughput assay. And then those 400 were then validated with a similar assay to what we were doing, surface plasmon resonance. And in the case where they were infilling only CDRH3, they observed a 10% binding rate of the 400 that they actually validated. So in our work, this isn't a direct head-to-head comparison. The purpose of including the Absci result is essentially just for context for those of you who maybe aren't as familiar with antibody design and kind of what binding rates you might expect. So what we did was we infilled 20 different lead molecules for 4 different antigen targets, up to a max of 8 model-selected positions. And instead of doing it in this one-shot fashion, as done in the Absci case, this was done over 3 rounds. So the library was adaptive in the sense that after each round of experimentation, we retrained the models and then repeated. And at the end of our 3rd round, we observed a 40% binding rate. So let's see, got about 5 minutes left, and then I will answer some questions. Right, so to kind of give you an overview, so we did this over 3 rounds. In total, we tested 374 sequences, 4 target antigens, 20 target-specific seed molecules. One of the interesting things that I want to point out is this dip in expression in round 2. And the reason that it's interesting is because essentially it was a result of not setting the coefficient on the KL term large enough. And so the optimization algorithm optimized too aggressively and resulted in low likelihood sequences that didn't express that well. And so then at the next round, we fixed the sequence length and increased the KL term, and the coefficient of the KL term and expression immediately recovered. So I think one of the big takeaways from this particular plot is the value of doing multiple rounds of experimentation, where essentially like the issue with the one-shot regime is that you're essentially just betting that the distribution of whatever data you were handed is like relevant enough to allow you to solve the task at hand. And I would say that's particularly when working with scientific data, rarely the case, because that scientific data was probably selected for some other reason with like extreme prejudice. For those of you who are curious, this is our these are the results of the yield measurements as a function of edit distance. And so one of the nice things to see here is that we essentially were able to produce sequences that express well across a range of edits out to around 10, which I would say is similar to some similar results found by a team at Google, led by Zelda Marriott, working on nanobody design. The other thing that's nice to see is that some of the sequences did actually improve the observed yield, although obviously the ones that are like close to zero, there's probably some observation noise here, and so you want to be careful about exactly how many. You'd have to run more replicates to determine exactly how many improved on average. And similarly, we have the same plot for binding affinity. Because more of the sequence is expressed than bound by necessity of the hierarchy. There's fewer points here, but again, we were able to find binders, in this case, out to 7 edits across a range of KDs. Although I think so basically ranging from micromolar to about nanomolar binder. Right. So the takeaways for this section, sequence only models are simple and easy to work with. Solving high dimensional black box optimization with deep learning really requires distributional constraints to make sure that you're not just exploiting your model. We're able to find sequences that are likely and score well, and ultimately this is kind of led to some encouraging early results that suggest that our method's effective for antibody lead optimization. I saw a bunch of questions in the chat, so I think I will.
Yeah. So Sam,
try to
I guess
get right to that.
one
Go ahead.
overarching one might be a question about how the positions are chosen.
Yeah. So I think I have. Oh yeah. Before I answer that, I just want to acknowledge the rest of our collaborators at NYU and Genentech. This was very much a group effort, and this paper would not be what it was without all their efforts. So in the paper, what we did was you compute this saliency map. Like I give you a budget of, let's say, 8 edits. You compute this saliency map, normalize it into a distribution using this temperature hyperparameter tau, and then sample a set of edit positions. So what you are doing is you're using the gradients of the discriminative head, not just to guide these token distributions, but also to select the edit positions automatically. Hopefully that addresses that question a bit more in depth. Let's see. Just looking at the chat here. Yeah, so Dasha asked, just to clarify, are you saying that you need to provide the exact mass positions in a sequence that are allowed to be modified? Can the model select them? So in the encoder only MLM style framework, yes, you have to the only positions that the model is trained to change are the ones that are masked. And then, yes, the model can select them by mechanisms such as the one I just described. Let's see. And then Jay says, once you have a discriminative model, can you use it to map the embedding space with respect to your function of interest, thereby finding regions of embedding space that are right for sequence generation? Yes, that's essentially exactly what we do. Oh, I see, Nate, you already answered that question. Did you find it useful to interpret the embedding space itself or describe it with respect to a function? I would say this is something that we're like still investigating. You can do like, Tisney plots, for example, to try to like cluster based on your embeddings, but we haven't like found anything like super useful to come out of that yet. Another one about edit position selection. I think I addressed that. Our cottage. How did your surrogate models perform in predicting properties of sequences with combinations of mutations where the individual mutations were not or only partially present in the training data? Well, the way that we did our split was essentially just based on sequence identity. So I think what you're referring to is people that will take like, one mer or two mer mutation data and then try to predict like former mutations. And that's not really the regime that we're in here, where like here we kind of assume we have multiple successive rounds to do, let's say like 8 edits. But I guess the takeaway here should be, if you're allowing the model to make like up to 8 edits, by necessity, the model is having to predict combinations of edits that the model of mutations that it's never seen before. But I think maybe to get at the spirit of your question, I would say like in our experience, it's really quite important that the mutations in your both the kind of starting point and mutations in your data is reflective of what will end up in your designs. And that's a big part of why doing multiple rounds of design and experimentation and training is so important. Edit positions are chosen independently and ignore correlations doesn't leave a lot on the table. Well, so I mean, I actually have So in this example, the samples from this normalized saliency map are drawn from a categorical distribution without replacement. So in that sense, they're independent. But there are pretty strong correlations in these saliency maps. And I would say one of the primary baselines, particularly like in genetic algorithms, is just uniform random edit position selection, which is which leaves even more on the table. So I think the short answer is that I'm sure that this probably is not the pinnacle of edit position selection. However, when we started the project, like essentially we wanted an alternative to hand selecting them, which is primarily what most people do when they choose like a CDR and just say change this, right? But that's not going to work once you start going into realms of like trying to optimize developability liabilities like tryptophan oxidation or aggregation. Okay. Before doing the 374, did you have these data for each antibody antigen system to train? So basically, the historical data that we had was not all for those targets or those seeds. It was kind of just like this large historical training corpus. And in fact, there was quite a severe distribution shift. So essentially, like the way you should think about it, it was like a historical database of experiments that scientists had done in the years leading up to it. And then we partnered with our antibody engineering department, and they gave us some targets and gave us some lead molecules and said, optimize these, really with like very little consideration for what was in the historical training data set. Right. So there's a question about the improvement. So the delta is the difference between the design and the seed, where the seed measurement was on the same plate or in the same batch. So at the very least, you're accounting for batch effects in this metric. You're exactly right that design, you know, basically values that are like very close to zero, you should probably run some replicate-SAR to verify that there actually was an improvement. In terms of the error of the experimental measurement, I mean, the short answer is that it's not homoscedastic, but the rule of thumb that we like to use is we like to look for a KD improvement of like 0.5. So like in this case, like for antibody engineering to be like really interested in a lead molecule, like this point up here around 5 edits out would be an example of that. And so if you're kind of thinking to yourself of like, okay, how much improvement should I be looking for like KD improvement to be meaningful, it would in this case be like half of a log 10 unit.
Okay, awesome. Thank you so much, Nate and Sam. I think we're almost at time. So if people have one or two last questions that they either want to unmute and ask or drop in the chat, we can stop after that. Okay, if not, we can go ahead and end there. Thanks again so much for the talk today. As a quick reminder, we'll have our last talk for the semester 2 weeks from now. Structure-informed language models are protein designers. So we'll see everyone then.
Thanks for your attention, everybody.